In this video, we will be talking about transcription. This is our DNA molecule. This region of it is the DNA coding region. This region is where all the information required for the construction of proteins is located. Just a little bit upstream of it, we have the promoter region, a -10 to -35 box. This region is also known as the Tata box because it is rich in thymine and adenine. The promoter region is what essentially makes a decision on when the initiation stage will start. The initiation stage is the first stage in transcription. At the beginning of the coding region, there's a start codon, and at the end of the coding region, there's a stop codon, which is the termination sequence. When transcription starts with the initiation stage, transcription factors, which are proteins, will bind to the promoter region of the DNA. These transcription factors will activate RNA polymerase, which will then bind to the promoter region. The number of transcription factors required for the activation of RNA polymerase depends on the gene. It could be any number, a couple hundred to only 4 or 5. For our example, we'll be using 3. Once the RNA polymerase is activated, the coding region of the DNA opens up and signals the beginning of stage 2, which is the elongation stage. In this stage, the RNA polymerase will move along an opened up strand of the DNA double helix to begin to make pre-mRNA. The RNA polymerase will move from the 3' to 5' direction of the DNA strand because it reads DNA from 3' to 5' in order to synthesize pre-mRNA. So the RNA polymerase will move along our 3' to 5' strand, which is this one, and synthesize pre-mRNA as it goes along. It will continue to synthesize pre-mRNA until it reaches the termination sequence, which is when the termination stage will begin. This is where the RNA polymerase falls off the DNA and the newly synthesized pre-mRNA strand is free. The mRNA strand is 5' to 3' because that is the direction it is synthesized in. In the new pre-mRNA, there are exons and introns. For our example, we will have 3 exons and 2 introns. There is no specific order for exons and introns. Before this pre-mRNA can become mRNA, the splicing must occur. This is when the introns are cut out of the pre-mRNA strand. To do this, spliceosomes will bind to the ends of the exons and the mRNA strand and create what are essentially loops out of the introns. These loops will then pinch off and cut the introns out of the pre-mRNA, which will give us our mRNA strand. We now have mRNA, which consists of only exons. However, before this mRNA is ready to leave the nucleus, it needs something to protect it from degradation. That would be a 5' cap, which is added to the 5' end, and a poly A tail, which is added to the 3' end. The 5' cap and the poly A tail protect the newly transcribed mRNA from degradation. It is now ready to leave the nucleus.